So we're ready for Rochester, New York, and this is a Mission 4 flight experiment. Let me also remind everybody that we have Mission 3A, 3B, and Mission 4 experiments, which have all flown, and we have Mission 5 and Mission 6 experiments, which are going to fly. They haven't flown yet.
All right, good morning, everybody. I won't need this because obviously you can see me. So my name is Cheyenne Jeffrey. My other co-principal and investigator, Vicki Ann, could not be here with us. I also have my chemistry teacher, Miss Mary Courtney, and I am from Rochester Early College International High School, recently graduated not too long ago, and I will be talking to you about tardigrades in space. Next slide, please. So let me go into about what are tardigrades. So tardigrades, or another term for tardigrades, which is water bears, is that they are microscopic organisms, and they can be found basically almost anywhere in the earth, which they mainly are found in moss, lichen, soil, water, and so on and so forth. Next slide, please. As I was about to talk about, these water bears are very known to be able to withstand or survive through in very intense conditions, such as you can see through, they could survive temperatures as low as negative 200 degrees Celsius and as high as 151 degrees. They can survive through lack of oxygen, water, boiling alcohol, and even high pressure. Next slide. So how are they able to do this? A little trick that they're able to do is called a technique called cryptobiosis, which is a temporary state where an organism metabolic activities come to a reversible standstill. So in other words, these water bears go into a temporary dormant state where they could where you can see in the second picture where they will shrivel up and they will go into the dormant sleep, and they would technically sleep through the extreme temperature until conditions until it's over, or have they been moved to a different area. Next slide. So for our experiment, we had conducted 2 experiments. The question that we had is there a structural and behavioral difference when tardigrades are in microgravity? So there were many studies saying that there were tardigrades found in space. So what we thought about is that, what about if tardigrades could do the cryptobiosis form in Earth, what would happen if they're reactivated back in space? Next slide, please. So what we had was the Type 3 FME mini lab capsule, and in the first volume we had the water bears that were dried up. We had dried them up, let them sit out of the water for a couple of days for them to completely dry out, which made them go into the cryptobiotic form. We had around 25 or so in that first volume. In the second volume, we had the hay infusion media with a few reed grains and Spirogyra algae, flamentis, that is what reactivate the water bears when they're being reactivated in space. And in the third capsule, we had live water bears in this, also with Spirogyra algae, and we had around 10 to 15. Next slide. So after the 6 weeks, we got the results, and they weren't the results that we thought we expected. We expected, a huge population, maybe different in sizes, colors, or something. But as you can see, there's kind of similar to each other. For the space rehydrated water bears, we couldn't find any signs of life. There was strong odor, cloudy, making it hard to identify. We were able to identify remains of a couple exoskeletons, so it may be possible that the water bears may have reproduced and may have leftover of its exoskeletons. There was a lack of algae, a yellowish color, and a pH level of 8. For the Earth's rehydrated water bears, it was about similar, strong odor, cloudy, identified less than 4. We were able to identify the the remains of 4 water bears, lack of algae, and pH level of 8 as well. Next slide. For our space ground truth, we were able to find exoskeletons as well. We also noticed there are other microorganisms such as rotifers, which we have not noticed from before. Also, lack of algae again. With the Earth's ground truth water bears, we had pH level of 7, which is around normal. Same order as the rehydrated experiment, and also same lack of algae, and making it difficult to see and identify less than for the water bears. Next slide. So when we were coming to our conclusion, we were trying to figure out how was it we were able to figure out what happened to the water bears, and we came across some clues of that there's a lack of algae and maybe the type of liver environment. For the lack of algae, it may have been possible that we did not have enough algae for the water bears, or we had too much water bears in the capsules for both two experiments. And also for the type of liver environment, we not for the water bears, they need to have moss. Even though they don't have algae as a food resource, they still need to have moss for them to even survive to do the cryptobiosis. So we came to the conclusion, even though that we were not able to answer our question, but we were able to, for future researchers, they'll be able to conduct a better experiment for the future. Next slide. Just want to make a quick acknowledgement for my financial support, the Rochester Area Community Foundation and the Asia Society International Studies School Network, and for any other support, my school, Rochester Early College International High School, the City School District, the Ward Science for my samples, and the University of Rochester for the microscope. Thank you very much.
Do we have any questions for the Rochester, New York team?
I'm Raquel from St. Jude Children's Research Hospital. And what would you do differently if you had to send this up again?
Differently, what we've done differently is that we may have conducted maybe one experiment with just the water bears that's being rehydrated in space, instead of having the live water bears and the rehydrated water bears. So I think that's probably what we would have done differently.
Do we have other questions?
My name is Christina Murray. I'm from Downingtown, Pennsylvania. Do you think the difference in pH between the ground test, which you said was 7, and the space test, which was 8, do you think that difference in pH would have any effect on the water bears?
We do believe that there may have been any effect with it. So maybe with more research, we will be able to figure out maybe what is the difference between those two.
Thank you.
I'm Nicole from Kansas City, Missouri. What made you think of this idea for...
Pardon?
What made you think of your idea for the flight experiment?
Well, at first we didn't know what to choose. We were looking at a book called the Ward Science book, and we just came across a moss piglet, which is also another term for water bears, and we were looking at it, and they were saying that how they were able to survive through almost any type of extreme conditions, and they were actually found in space. So we decided to challenge that, to see, okay, well, what happens if we reactivated them back in space, and then we went on from there and so on so forth.
Thank you.
Hi, I'm Elizabeth from Louisville, Kentucky. I just wanted to ask, how did you ensure that they were in the cryptobiotic state? How are you positive that they were in that state?
Well, if you had seen from the pictures from before with the definition of the cryptobiosis form, that what we were looking for is, if you could see it there, if we see the water bears shriveled up, then it means that they're in the cryptobiosis form. So we were able to identify a couple of those. They were in that form. So that's when we were completely sure and confirmed that they were in the cryptobiotic form.
Thank you.
Hi, I'm Harshit Sani from Melbourne, Florida. Mm-hmm. And I was wondering, you said that these were found in space. Could you elaborate on that?
Well, they were when we were researching, they were I believe they were saying that that certain people were investigating about the water bears, and they may have sent some in space. So apparently those had gotten lost in space, so they don't know what had happened to it. So I don't know how they were able to find the water bears in space, but they were saying that they were able to identify in certain vacuums of the space, that they were able to identify a certain amount of them, and they were actually alive and reproducing.
Thank you very much.
Thank you.
Great job. You had a lot of questions. Let's thank Rochester, New York.